So good afternoon, everyone. From Luca Segantini, I'm the CEO of ESOT. I'm very pleased to welcome you all to this webinar. Promises to be very exciting. My task today is only to give you a couple of technical tips and information before we start. Since we have 370 people registered for this webinar, we cannot, of course, keep everyone with their microphones on, which is a pity. But there will be a very nice question and answer session at the end where you will be allowed to send your questions for the speakers and the chairs and interact with them. You can do that in two ways. You can either use the hand button that you have on your GoToWebinar interface, and we will see that you raise your hand, and the chairs will select by priority and unmute your microphone so you can speak up your question and listen to the answer. You can also type your question in the question box, and we will see the questions coming in, and in the same way we will select them and ask the speakers to answer them. Other than that, I wish you a very good experience today, and I'll leave the floor to Professor Lorna Marcin and Professor Stefan Schneeberger. Enjoy.
Thank you, Luca. My name is Stefan Schneeberger. I'm a transplant and hepatobiliary surgeon from Innsbruck in Austria. It's my distinct privilege to co-chair this webinar together with Lorna Marcin. The title of this webinar is Prepping the Kidney Transplants of the Future, and it is part of Working Stream 1, leading up to the Transplant Learning Journey, an event that will take place later during this year. So this series of webinars is preparing the delegates and the community to the thought process and the principles of these working streams. Now, the core of the interest of Working Stream 1 is developing new endpoints, new targets for the emerging technologies such as bioenergetic preparation and generation of kidneys, but also xenotransplantation. So for these emerging disciplines that will eventually help hopefully to solve the organ shortage, we need to reconsider the traditional endpoints in order for that to fit the purpose. So I'm welcoming everybody to this webinar. It's certainly a high-profile webinar with a number of very good speakers. I'd like to also invite people to send questions or participate actively by raising a hand through the webinar. Without further ado, I'll pass it on to Lorna, also in order to explain how xenotransplantations will eventually connect to the development of new endpoints.
Great, Stefan, thank you very much indeed. My name is Lorna Marcin. I'm a professor of transplant surgery at the University of Edinburgh and immediate past president of the British Transplantation Society. And like Stefan, it is a great pleasure to welcome you to this webinar, which I think it will be exciting and fascinating. We have some excellent speakers, each of whom will deliver 10-minute talks, and we will keep strictly to time because clearly we only have 1 hour for this. And as Stefan really has highlighted, although xenotransplantation in itself is of considerable interest to us, we'd like you to focus during this webinar on really thinking about good examples of relevant and key endpoints. How do we define success with these new interventions that we're describing, of which xenotransplantation is one? It would be just as relevant in the context of regenerative medicine or novel technologies. So it's really how do we define success with these new innovations? So we'd like you to bear that in mind as you listen to our 4 speakers and to think about, please do contribute to the question and answer session at the end. So without further ado, it gives me great pleasure to invite Professor Robin Pearson to deliver his talk on when will xenotransplantation be called a success. Robin is Chair of Cardiac Surgery and Scientific Director for transplantation science at the Massachusetts General Hospital in Boston and professor of surgery at Harvard. Robin, it is a great pleasure and honor to have you here, and we look forward to hearing your talk. Thank you very much indeed.
Thank you very much, Dr. Marston, Dr. Schneeberger, and welcome to the audience. My task today is to help you understand what, at least I believe, how we will define success for xenotransplantation. Next slide, please. Next. So first, let me start by introducing the notion of why we're interested in xenotransplantation. As all of you will well understand, the supply of organ allografts is simply never going to meet the demand for life-saving organ and cell transplants. So one of the advantages potentially of the xenograft is that we can define the donor ahead of time and qualify the organs before they are released for transplantation. Those organs will come from a normal, healthy animal, not from the circumstances of cytokine storm and tissue injury associated with brain death. We can also avoid the transmissible diseases like hepatitis and other infectious complications that we spend a lot of our time in allotransplantation worrying about, because we'll source these animals from specific pathogen-free facilities. And Dr. Scobie will be talking about that in her presentation. The supply of organs is potentially unlimited, and particularly because pigs have very short gestation, 3 months, 3 weeks, and 3 days. They grow rapidly to adult size, and they have large litters. The supply of pigs of a desirable phenotype and genotype can be expanded to meet the needs of the human population. The organ's available when the patient needs it. It also, knowing the donor and the recipient ahead of time gives you an opportunity to condition both in order to optimize the chances of success and even potentially to induce tolerance. Next slide, please. So the question then of when to go ahead with transplantation of organs from pigs into humans depends very much on the results of preclinical trials. Preclinical xenoqualifying trials, two sets of examples are given in the lower images on the slide. And the red curves at the top of each slide illustrate that for the heart in the lower left, as of 2016, really great progress had been made relative to prior efforts. And this was based on genetic modifications to the pig and adding in drug treatments and other conditioning regimens to address identified problems that were encountered when pig organs were transplanted into non-human primates, either baboons or monkeys. On the lower right, you can see that there are 3 approaches that have been tried for kidneys that reliably yield success out past 6 months and often for past a year, which is truly a remarkable accomplishment considering that just 5 or 6 years ago, the longest survivor was not yet to 90 days. So not illustrated here, great results have also been obtained for islet xenografts and for neuroendocrine grafts. So what is the preclinical model has never been tested in terms of its predictive ability to guarantee results in the clinic. Nonetheless, it's our best available predictor. Next slide, please. So in Dr. Reichert's talk, which will follow, he will go through in some detail what the results have been achieved in his orthotopic heart xenotransplantation model. But just to bring any of you who are not aware of this work up to speed, this was truly a landmark achievement, reproducibly able to support the life of a baboon recipient for up to 6 months. And this work has recently been extended, as I'm sure Bruno will share with you. So next slide, please. Importantly, that result was achieved with a tolerable regimen, healthy animals at the time of elective experimental termination. So when should we go forward with preclinical trials? In the spirit of this webinar, I've been asked to consider what endpoints we ought to look at to decide when to go forward. And these should be identified both in the preclinical studies and then as we're designing the clinical trial itself. And as with other clinical trial benchmarks, non-inferiority is an important concept and one that I think should be the basis for coming forward with xenotransplantation trials. So when the preclinical studies predict that the outcome of a clinical effort will be comparable to other options that are available for a given patient, that is when I believe xenotransplantation should be considered for coming into the clinic. And that's because that prediction is the basis for ethical equipoise in presenting a patient with their options, in obtaining truly informed consent, not only from the patient, but in the instance of xenotransplantation, where there are unknown risks and potential risks, not just to the patient, but to those who live with them or care for them. This process is quite important, and good literature exists for those who are interested in pursuing it further. In addition, this ethical equipoise will be essential in order to persuade regulators, both institutional IRBs and national health authorities, that the time is indeed right for a clinical trial to go forward. Next slide, please. So in addition to non-inferiority, one of the important features of any clinical trial is to be sure that the approach that's being tested is likely to be safe. And the preclinical models give us good indications of whether infectious risks or drug side effects or other undesirable complications such as promoting malignancy are likely to be observed. And one of the very reassuring aspects of the Munich orthotopic heart xeno trial that you will hear about in the next talk is that there was no evidence of xenospecific endogenous retrovirus infection, for example. There were no unknown unknowns that cropped up in the clinical experience to date. And the drugs involved in that trial were either clinically approved or unlikely to be associated with serious side effects or malignancy. Next slide, please. So from that perspective, the Munich experience was very reassuring with respect to safety. And of course, this work will need to be replicated and carried forward to define whether it is likely to be successful. So how would I define success for a xenotransplantation? I would submit that an outcome should be expected to be similar to allotransplantation, that a recipient of a xenograft could with confidence expect to return to an independent functional existence, that the regimen would be tolerable with manageable side effects, and that their longevity with this intervention would be comparable to what we expect these days from a successful allotransplant with a good quality of life. Next slide, please. So while we cannot say with confidence that the preclinical models are going to be entirely predictive, we should expect that to benchmark our clinical results against both what happens in the preclinical models, because this is, after all, the best available way we have of gauging the likely success of our approach. Second, we ought to consider the xenotransplant against alternative therapies. And for patients such as those with mechanical heart valves who don't really have a good mechanical support option to transplantation, and especially those who are highly sensitized to alloantigens, as Dr. Kotze will talk about in his talk, there are groups of patients for hearts, for kidneys, for other organs who are very unlikely to either get an allograft or to benefit from alternative support treatments. For those patients, ethical equipoise will identify the patient population and expected endpoints. Infection control should be predictable, and Dr. Scobie will touch on that subject. I would urge all to approach xenotransplantation understanding that we can expect a learning curve, that we may not achieve success with the first efforts, but the study should be designed so that we learn from unexpected adverse outcomes and learn to accomplish success building upon our knowledge of the problems encountered. A good clinical trial, of course, will depend on thorough education, a transparent trial design, and proceeding deliberately, not under emergent circumstances, but with fully informed consent, not only from the subject, but from their close contacts. Next slide, please. So success in xenotransplantation, I would submit, is coming sooner than you may have thought possible. And the following speakers, Dr. Reichert, Dr. Scobie, and Dr. Kotze, will help you to see why I think that this is true. Thank you very much for your attention, and we'll defer questions, I guess, to the end.
Thank you, Dr. Pearson. This was a striking beginning. I think you perfectly set the stage for the next speakers to follow up. You already touched on the work from our next speaker, Bruno Reichert. He's now Professor Emeritus of Cardiovascular Surgery at the University of Munich, the LMU, and he's a co-speaker of the German xenotransplantation program as funded by the German Research Foundation. You have already briefly touched on his extremely important work that was published in Nature just a while ago. So without further ado, I'd like to ask Bruno Reichert to deliver his presentation.
Okay, well, we are a research foundation in Germany. We're altogether 3 universities share and are in control together with the German Primate Center, the Paul Ehrlich and Robert Koch Institutes. Altogether there are 18 programs, including ethical issues. The next slide, please. That's my talk. Xenotransplantation for heart failure, how close are we? And I have nothing to disclose. Okay. If you want to be successful, preclinically and clinically, you need to solve prerequisites. You need stable genetic modifications in the donor animals to avoid, that's the first line, xenoantigens and preferred antibodies. And you do that by knocking out major porcine antigens. For baboons, only the first one, the gall knockout, is important. Second, you need to stop the activation of the complement system. And then in yellow, you add human complement regulator proteins. Probably 1 is enough, but 3 probably is more. We don't know. The peaks were on our Earth approximately 80 million years before we appeared. So there are coagulation incompatibilities. And if you don't want to have thrombotic microvasculature, then you have to add human thrombomodeling. Then fourth, there are unspecific inflammation reactions, and you can add A20, H01, or human CD47 to block the monocytes. And the line on the bottom, later you have to stop the adaptive immune system, and you add anticoagulation antibodies is expressed on the program death ligand, or you knock out the swine leukocyte antigens that's corresponding to the HLA in humans. Next slide, please. Now we have our animals in a specified pathogen-free breeding facility, and that is led by Eckhard Wolf, who is our bioengineer. Next slide, please. And the second one, what you need, part 2 you need, is the clinically acceptable immunosuppressive regimen. And the mainstay is a co-stimulation blockade, as Mohamed Mohyeddin, who was already mentioned, taught us. He used anti-CD40 antibody, and he achieved a median survival of 298 days in 5 animals. The longest was almost 3 years, and he's then discontinued immunosuppression. Maybe the hearts will still be beating. Next, please. Unfortunately, he used heterotopic abdominal heart transplantation, which means the hearts were connected to the abdominal aorta and to the inferior vena cava. So the hearts were beating, but they're not included in the circulation. So it was an immunological model. Next slide. So before you enter the human, you have to have a consecutive life-supporting heart transplantation model. And you need success in 6 out of 10 successful cases for at least 3 months. That was set up in 2000 by David Cooper. Next slide, please. Now, the ultimate model would be Hodge transplantation, Hodge replacement. So they take out the baboon heart and replace it with a genetically modified porcine heart. Next slide, please. 3, the clinically approved heart preservation techniques don't work, obviously, in pigs. So we introduced in cooperation with 16 from Lund University non-ischemic heart preservation, which means the heart is taken out and all the time perfused, even during implantation. And so the solution is 8 centigrades cooled. It's a hypercortic solution. Hormones are added, nutrients are added, and hormones. Next slide, please. Now, size matters. The heart of a pig grows fast as the pig itself grows within 6 months. This is in contradistinction to a baboon. A baboon grows up slowly within 10 years. And the organs in a pig are genetically determined to grow fast and fast, so they overgrow the interior, the thorax, the chest of a baboon. Next slide. You have to inhibit overgrowth by lowering the blood pressure. Baboons have a higher blood pressure than pigs, and we wean cortisone early, and most importantly, as I think, we added rapamycin. This is a growth hormone blocker, a universal growth blocker hormone. For the clinical use, smaller donor pigs will be advantageous. Next slide, please. This is one of our pigs which are available there from New Zealand and Ireland, south of New Zealand, Auckland Island, and from nature, due to they were adopted the climate over the very cold over there. They grow only very small. They will become ultimately 70 kilograms. And our new Auckland Island pigs are porcine endogenous virus C free. Next slide, please. So this is in summary our latest results. We were able to be successful in the long term, at least for 3 months, for 6 months, in a consecutive series of 8 animals. We lost 2 in red due to porcine cytomegalovirus, and if you use porcine cytomegalovirus negative animals, then you will have no problem. Next slide, please. These are the hearts. Pigs were in good health when we euthanized the recipients. The baboons were in good health when we euthanized them. And you see the hearts are more or less, to the left is the left heart, to the right is the right heart. Beautiful shapes, anatomy in red. These are the 6 months hearts. Next, please. This is my group. And next slide. So xeno heart and kidney transplantation, it is time for a clinical trial. And next, please. That we think it should be. You need for Europe 1 farm, and you take out the hearts or the kidneys and fly them or drive them to the recipient hospital, where the physicians, the surgeons do then the intervention, and that's it. That's our plan, how we'll do that. Thank you very much for your attention.
Wonderful. Thank you very much indeed, Professor Reichart, for a really fascinating description of really all of the work that you've done in very practical ways of how you've overcome some of the challenges. And we're having already some interesting questions coming through, which we look forward to discussing with you at the end. It now gives me great pleasure to invite Professor Linda Scobie to deliver her talk, who works in Glasgow. So not very far from me, but we could be anywhere on these webinars. But Linda works a full-time lecturer and researcher at Glasgow Caledonian University in virology, particularly with respect to zoonotic transmission, and really works around providing supporting safety data to move xenotransplantation to the clinic. So we really look forward to what you have to say, Linda, around the key regulatory information to make this happen. Thank you very much indeed.
Thank you to the chairs. Thank you to ESOT for the kind invitation to give some key regulatory points on xenotransplantation and taking this forward to the clinic. I have no conflict of interest, and here is my presenter disclosures. Next slide, please. I guess the first question is why is it necessary? Well, just to put it in context, allotransplantation with human organs is not considered what we call a medicinal product in either Europe or in the United States, and therefore no regulation is required. However, for xenotransplantation, the regulatory path to clinical development is a bit more complex. And this is due to the need for the intended use to be significant. And as Dr. Pearson mentioned early on, there are targets to be met with respect to safety, efficacy, amongst others. Indeed, xenotransplantation is considered a medicinal product and is under regulatory oversight by the European Medicines Agency in Europe and the FDA in the US. Next slide, please. This is a timely talk as myself and Dr. Henk-Jan Schuurman have just guest edited a thematic issue in the Journal of Xenotransplantation, which has had input from global experts in experience with clinical trials we've carried out, and also other aspects which are really important. So I'll be referring to a lot of the manuscripts published in this issue throughout the talk. Next slide, please. Okay, so regulatory pathways are governed in Europe by the Medicines Agency, and as I've already said, in the US by the FDA. You need to approach these regulatory bodies in order to gain approval for any clinical trial. So for example, in Europe, your application would be discussed and submitted via the European Medicines Agencies and or the national competent authority. Once you've engaged with the EMA, you then need to complete what we call a clinical trial application. This might need to be accompanied by what we call an investigational medicinal product dossier, and I'll come to explain more as to why this is necessary as we go further in the talk. These documents are submitted through a portal, and timelines, if there's no issues with your application, is usually about 45 to 60 days. It can go up to about 106 days if there are issues, and again, this is in line with the data you present with them. Likewise, in the United States, there is also a similar process via the FDA, who are involved in the oversight of any clinical trials, and interaction will occur with various committees depending on how your product is identified. This may also involve completion of what we call a new animal drug application, or NADA, or indeed an investigational new animal drug, or an INAD. This then goes on to be reviewed and viewed by the Institutional Regulatory Board. You can see there's quite a lot of committees involved in this. Next slide, please. So what do the regulators want to know? Well, I think the first and most important thing in order to help you decide where you seek approval and what guidances are important is that you need to know the definition of a xenotransplantation product. These definitions differ between Europe and the United States, and you have to be clear on what those definitions are. On the left is a list of some of the IDs of how xenotransplants are defined, and this is not exhaustive. Next slide, please. So this is taken from the thematic issue I mentioned earlier by Godehart and Tonjes, and here they define the classification of xenotransplants. So what's quite important in the EU, an organ would be a medicinal product or xenotransplant. However, if this is then genetically modified, it becomes what we know as an advanced therapy medicinal product, which requires additional guidance and additional directives to be adhered to when you submit your approval. There are also issues as to whether the product is acellular, or whether the product will still maintain what we call substance, and that means there are some elements of what was originally there in the porcine-derived product. So, for example, acellular, they would be treated with glutaraldehyde, so there would be no cells left, etc. However, there are other products where you can still detect DNA, and again, these would be considered differently. So again, what we have here is the definition between a medicinal product and then a medical device. This then again differs when you go to the US, whereas a decellularized or an acellular product in the EU is called a medical device, or if it's in combination, for example, islets with an alginate surface, then it becomes what we call an ATMP. However, in the US, a decellularized product is a xenograft, and an acellular product with some components remaining, they call a combination product. So you have to be really careful how you define these. Next slide, please. So as I've said, defining your product to the regulator is really important, and that will guide you to the relevant authority and the appropriate approval process. The box on the right, considerations for xenotransplantation trials, again, it's really important to take all aspects into consideration. You need to think about the source herd, the source animal, processing, procurement, and then also the recipient. The key thing here is good practice and good documentation. So good regulatory practices do apply. There should be full detail, tracking and traceability of the herd, safety screening protocols in place, which we don't have a lot of time to go into here, but you'll find information in that thematic issue. Specific husbandry is applied to select animals which are designated pathogen-free. And PERV analysis, or porcine endogenous retroviruses, is a mandatory part of screening at this point and also at the end point, regardless of whether you consider PERV a risk or not. At the end point, it will still have to be done from guidances in both the US and in Europe. As mentioned earlier, harvest and transport is very important. The organ must be controlled and in a sterile environment. Again, traceability, quality assurance, and documentation are key. Next slide, please. So this is just to summarise. Data management, data collection, again, I can't emphasise how important that is. The more information you have and the more you present to the regulatory body, the easier it will be for them to understand your management system and also identify gaps where they may require you to go and find out some more information. So this flow chart just essentially describes the process of gathering that documentation and providing evidence which you will then take to the regulatory body. One link I'd like to point out is that the WHO and the International Xenotransplantation Authority are very much involved with the Transplant Society in developing and reporting what we call the Changsha Communique. In 2019, there was an update on this, and again, there's lots of information in this as to how to proceed with your regulation. Next slide, please. So in summary, advice, interact, early conversations between the sponsors and the regulatory authority is really important. The focus from the regulatory perspective is not about the techniques you use, it's about product. You must ensure all evidence is documented and provided, and therefore regulation should actually not be a barrier to moving forward to the clinic. Next slide, please. Just as an example, the FDA has this meeting called Interact, and this is where you can engage with the FDA prior to submitting any application to get as much advice and feedback as possible, and they're very happy to do so. Next slide, please. So where do you find the information? As I say, depending on the product you select, there are different guidances, regulations, directives, etc. that you will need to refer to in your application. And in the next slide, I've listed some key links that you might find useful to go and look at. Specifically, if I could refer you to the document in the thematic issue by Sherman and Hubendorn, which is on solid organ xenotransplantation at the interface between research and clinical development. Last comment I'd like to make is that solid organs are going to be much more complex regarding clinical approval, mainly due to the fact that there isn't really a huge regulatory framework in place for this. It's much easier to take xenogenic cellular products forward because we already have regulatory frameworks in place for human-derived cellular products. So with that framework already being established, it's much easier to integrate xenogenic cellular products. So there may be some more time in order to get by with the approvals for solid organ. Thank you for your attention.
Thank you, Dr. Scobie. This was a great summary illustrating the enormous effort that would be required for xenotransplantation to advance to clinical trials. For a non-expert in xenotransplantation like myself, this is very helpful to understand the complexity and the dimensions of the efforts to better also understand why this is not advancing more rapidly. Now, without further ado, I'd like to introduce our last speaker in this panel. Emmanuelle Cozzi is a clinical immunologist with expertise in the field of transplantation, and more specifically, his focus is on the role of antibodies in transplant rejection, both HLA and non-HLA antibodies. Emmanuelle has extensive know-how in pig-to-primate xenotransplantation and is also the past president of the International Xenotransplantation Association. Emmanuelle, we're greatly looking forward to your talk.
Thank you very much, Stefan. I would like to thank ESOT for inviting me for this very interesting webinar. Here are my disclosures. We can move on, please. Okay, so basically, I had been asked to speak about the patient selection for clinical xenotransplantation trials, and more specifically, to focus a little bit on the kidney. And the question that I was asked to address was, which patient should be tested first? Shall we go ahead first with highly sensitized patients? Shall we go for other groups, or is there a case to consider low-risk patients at all? So I will take you through a few slides that will explain you why I definitely selected this hypothesis to move forward, paying particular attention to highly sensitized patients. If I can have my next slides, please. Yeah, so basically here are two different situations. I have selected the situation in the United States as reported in the latest report of APTN and SRTR just published, and also the situation in Italy. What we can see here is that basically we do have this green population and this brown population. These are the hyperimmunized patients, and in particular, as you can see here, we have patients who have a calculated PRA, very severely hyperimmunization. And as you can see, the trend throughout the last 10 years and hasn't changed. So basically, these are really patients who are unfortunately severely hyperimmunized, and they are stuck. They don't seem to have the availability of going ahead with a transplant. And as far as Italy, we do have a similar scenario. And a year ago, we had approximately 17% of the patients on the active waiting list who had a calculated PRA greater than 90%. And amongst these, 1 in 3 was not only hyperimmunized with a calculated PRA of at least 90%, but they had been on dialysis for longer than 8 years. So we really do have a major problem to provide access and opportunities to transplantation also to these patients. The next slide, please. So now, what can we offer to the patient hyperimmunized today? Unfortunately, the slide is slightly unclear, but it's not a problem. Certainly, we do have national programs around across Europe, across Italy, and across the United States. We can certainly think about desensitization, and then there are very peculiar programs like the acceptable mismatch program, for instance, if you in Eurotransplant. Now, there are important things to consider, though, that we know today that if we take hyperimmunized patients to transplantation, we have to accept that here, this is an American study, and it shows the percentage of mortality. We have to accept that severely hyperimmunized patients, they will certainly have a mortality, which is 2 to 3-fold the mortality at 5 years that is expected if the patient is non-immunized, and that's the continuous black line, okay? Now, on the other hand, if we look at the data that have been provided by the Acceptable Mismatch Program, certainly here the situation at 10 years is quite good in terms of graft survival compared to non-hyperimmunized patients. This is the green line. It's slightly lower, but it's still an excellent survival. But the key point of this slide is that the patient who can have access to these very privileged programs are very, very, very little numbers, very little figures. We are talking about 800 patients versus 16,000, 17,000 patients who are immunized waiting for a transplant. So these programs, they are extremely good in terms of outcome, the Acceptable Mismatch Program, but unfortunately, they are not programs that can be offered to all hyperimmunized patients. Can I have the next slide? Now, when we talk about xenotransplantation, indeed, we are in an extremely privileged situation because we do have the genome editing. We have been hearing speakers about it before. We can really tailor the potential donor for many aspects to render it more acceptable to men. Of course, the first few things we are talking about is about immunological disparities and to try to calm down the immune system, complement regulator, changing the antigenic profile, as we have heard before. But we can also work on physiology with genetic engineering. And more importantly, today, we can work also on safety aspects because the tools we have at hand, as you know, we hear more and more talking about the Zinger finger nucleases, TALEN, CRISPR, Cas9 technologies that really enable us to customize an organ, a donor and an organ with knock-in, knockout, and also transgenic approaches that would render really a pig organ more compatible with man. The next slide, please. And here I'll show you that, as we have heard before, the results around are really very good. This is the best results that have been reported. And I would like, and this is renal xenotransplantation, life-supporting organ. The key aspect I would like to bring to your attention is that here, working both with an induction treatment with anti-CD4 and also anti-CD4 and also maintenance with anti-CD154 monoclonal antibodies, in addition to solumedrol and mycophenolate mofetil, we can really have exceptional survival. I mean, really, we do have reproducibly survival longer than 1 year. And it is notable to notice that the donor profile, from an immunological standpoint, these are not the best engineered pigs we have. We have better, and still we can reach more than 1 year survival in these primates. And incidentally, these primates were also selected because apparently they had low titer of anti-pig antibody. The next slide, please. In all cases, the important thing is that the antigenic profile of these animals today are dramatically different. This is an example of a patient's serum that is tested either on a wild type cell line, okay, and then you see a tremendous binding. But then we move on with the genetic engineering. We arrive to a stage where we have a triple knockout pig, without going into the details, where 30 to 40% of the sera are no longer reacting with these sera, okay, with these cell lines. The next slide, please. Now studies have been hold on, there is something. Well, studies have been ongoing, and these tests have been trying to use also the kits that test donor-specific antibody in clinical transplantation. There have been very interesting experiments produced using sera from hyperimmunized patients. If we go ahead, the next slide, please. What we can see is that indeed this is a a patient an intact serum, and as you see, there is a high binding. This patient is hyperimmunized against MHC class 1 antigen, and we have very high NFI. Okay, what we can see is that sera that are eluted from human cells, okay, in fact, we can definitely reduce the binding of these sera to the beads of the Luminex platform. But the important thing is that what we have to know is that indeed we have evidences that porcine MHC molecule class 1, SLA class 1 molecules do indeed share some epitope with MHC HLA class 1 antigen. Can we go ahead? Next slide, please. And the interesting thing is that there have been studies conducted with patients from hyperimmunized patients, and what we can see is that the binding of these sera to GAL knockout cells, okay, is much higher than the binding of these sera. These are 18 patients. The binding to these sera is dramatically reduced if we are not exposing the serum to pig cells knockout for GAL knockout, but also knockout for the MHC class 1 molecule in the pig. And the same thing, can we move on? The next slide. Same studies are underway for the studying the SLA class 2 antigens, where indeed there is evidence that there is cross-reactivity between sera that recognize HLA molecules and will potentially cross-react in 60% of cases with some pig SLA class 2 molecule. The next and last slide is somehow the take-home messages, really the concluding remarks. So unquestionably, 10% of the patients on the waiting list are very severely hyperimmunized, and we know that there are about 10% of the patients who have a CPRA greater than 98%. They will be unable to accept 98% of the potential donors. This patient will wait a very, very long time, notwithstanding the programs that may exist and are around for a few and not to all the patients, okay? As I showed you, we can now reproducibly obtain long-term survival of primate recipient of kidney xenograft, and we have seen the outstanding results in Munich obtained with life-supporting heart. So really the results are very good. And the proposed immunosuppressive strategy seems something that should not tremendously increase the infectious risk of our patient, okay? So clearly, in our view, xenotransplantation may represent an opportunity to hyperimmunize patients, but clearly we know that there may be some cross-reactivity between anti-HLA antibodies and HLA class 1 or 2 antigens on pig cells. So therefore, studies are underway, and indeed we believe that we have a very strong plus, not only for immunology, as I said, but as far as immunology, the genome editing will certainly provide us much new lines of transgenic pig, which are much less immunogenic, and they will probably represent an opportunity for successful xenotransplantation in hyperimmunized patients. And I stop here, and I thank you for the attention.
Thank you, Emmanuelle. This was another great summary addressing now kidney xenotransplantation more specifically and pointing out some of the other challenges ahead. May we ask all the panelists to switch on their cameras and microphones for the discussion. Thank you to the presenters for staying on time, each and everybody, so that leaves us with the predicted 10 minutes time for discussion. As a reminder, there's two ways of submitting questions. One is typing in the questions through GoToWebinar, and the other option is to raise your hand. I'll pass it on to Lorna to introduce the first question.
Okay, that's great. Thank you very much indeed. I'd like to echo Stefan's thanks to the speakers for very thought-provoking talks. So I think an interesting question that actually was the first question asked on the chat was around how are we as a community making sure that the patient voice is being incorporated as we discuss defining endpoints, and success in the concept of xenotransplantation. I wonder whether, Robin, you might start the answer for that. Thank you very much.
Thank you. It's an excellent question. Among the approaches that the Korean group, who are moving forward with cornea and islet transplant, and the German group have taken, as you've heard, Bruno and his team are looking at hearts and probably kidneys as well, is to engage the local communities, both the professional communities and the lay communities, in a dialogue about the promise and the risks associated with xenotransplantation. And those dialogues have yielded good back and forth communication. Very often, as a result of that communication, the acceptance of xenotransplantation is improved in the population who have been engaged. That said, it is difficult to get the attention of the population at large, and there'll be, it is a challenge that remains before the field to know what the best practice ought to be in that regard.
That's an extremely important aspect, as you pointed out. Also, ESOT is engaging in patient communities in order to address such questions and involve them more intimately in the discussions that we have with the EMEA. I think that can not be overestimated. Now we have a question from James Newberger. Thanks, James, for joining us. It's a privilege to have you with us. And his question is, can the speakers comment on the physiological aspects of xeno, for example, porcine livers, make porcine proteins, and so on? What effect will that have on humans? Maybe, Emmanuelle, you can briefly address that aspect of kidney, maybe even liver transplantation. Do they produce the same proteins? Is there sort of another barrier awaiting us with respect to the differences of the synthetic function of these organs?
Yeah, so as far as liver, definitely the physiology is quite complex, and we are aware that there are substantial differences that, including certainly the production of complement factors, that render liver xenotransplantation a real challenge. And we know, by the way, that pig to primate liver xenotransplantation has not, as yet, produced the good results that we have obtained so far with cardiac and kidney transplantation. So we don't even reach a month as yet, but they are working hard and they are improving in terms of survival. In terms of kidney, I mean, for sure, and heart, and I won't take the floor, and I'll let certainly Bruno comment, but as far as kidney, for sure, these animals, the porcine kidney is life supporting because it's a bilaterally nephrectomized primate, and it goes on forever, at least for more than a year, and we don't seem to have major problems. So as far as we are concerned, I mean, so far, physiology is not an issue for long-term survival of renal xenograft recipient, for a renal xenograft recipient.
Maybe building on this question, I guess what everybody has in mind and what people keep asking for, Bruno and maybe Linda, is when will we see it happen, considering the regulatory burdens and the regulatory limitations, but also the sort of the medical incompleteness of the understanding of xenotransplantation. People have been talking about this for a long period of time now. When will we see the first clinical trials?
Well, obviously there have been clinical trials using islet cells, New Zealand, Argentina, amongst other places. In the thematic issue, there is some nice paper by Sean Wynyard describing the New Zealand experience and the trials and tribulations they faced, particularly with maintaining archives of samples from these patients. I think it's important to point out that to date, regardless of all of these clinical trials that actually have taken place, none of them yet have demonstrated any evidence of adverse events or indeed exogenous or endogenous retroviral transmission and evidence of infection. So realistically, we are in a bit of a catch-22 where we need to get trials ongoing to definitively prove that this is what we see is no longer an issue. So without getting the approvals and getting the trials done, we can't really demonstrate and prove this that there is no issue. So we're a bit stuck with that.
I'd like to thank all of the speakers for really excellent talks, for keeping so well to time. And I think in many ways, one of the real is that Robin's comment that we're closer than we think. And yes, there are regulatory challenges, and it's fantastic that we have individuals like Linda who are really engaging with us about how we can overcome those challenges, which are obviously different in different parts of the world, and the revolution that is genome editing that Emmanuelle described so well. So I'll just hand over to my co-chair to close the webinar. Thank you very much indeed.
Thank you, Lorna. I would like to second what you just said. Thank you for all the speakers for keeping the time. The recordings of this webinar will be on our ESOT Transplant Live platform within the next several days. Thank you for joining us. I hope you will participate again in the upcoming ESOT webinars. We'll continue certainly this series. Thanks to all the panelists for excellent talks. I think you summarized this chapter extremely well. Thank you, Lorna, for co-chairing this with me. Thanks to all the delegates. Thanks to the ESOT staff for organizing this webinar so well. Hope to see you again sometime soon on the next ESOT webinar. Goodbye.
Thank you very much.